Thanks, Neil. Thanks very much for the introduction. Yeah, so my main interest, as Neil said, in kind of drug discovery for motor neuron disease. And I'm going to try and cover kind of the kind of regulatory framework that we work under, which is a legal framework that imposes certain ethical considerations on us and kind of a requirement to have optimal experimental design. Now, as Neil said, I'm not a computationalist, in one sense, kind of the data that I'm showing here, there may be, you know, more advanced ways of analysing what we're doing. I'd be really interested in people's opinions on that as well. So I'm just going to go through how we approach these issues. So I'm going to start with a bit on the ethics of animal research, why we use animals in research, then talk about the law and the regulations we actually work on, and what that means for us, but practically, day-to-day, running experiments in mouse models. And I'll then talk about mouse models of motor neuron disease, and some of our data show you some of the approaches we've taken to try and kind of minimize the number of animals we use, minimize the distress that we cause when we're trying to test new therapies, and so on. But to do that, I need to give you a bit of background, I think, on motor neuron disease. So hopefully a slightly different talk to the ones you've been exposed to so far. Okay, so very simply, why do we need animals in biomedical research? I guess we should start from that position. You may not all agree that we do need to use animals in research. Certainly not everybody working on biomedical research would, though the vast majority would. And from my perspective, I think it's impossible to model intact physiological systems, either in vitro or in silico. I think there's certain things we can do well. We can model certain aspects of biology, but we can't model an entire intact physiological system. We have to go into that system at some point, conduct an experiment, and see if our models that we may be using, whether they are in vitro or kind of in silico, are accurate or not. And just thinking about two systems that I know a bit about, so the immune system, the nervous system. If you begin to think about those at all on a biological level, it really is impossible to do that in anything other than a kind of intact animal. And even if it were possible, we would still want to confirm any model predictions in the real system. So there's certainly possibilities for modelling to reduce kind of non-animal methods to reduce the number of animals that we use, refine our experimental interventions, but at the moment we still need to use animal models. And broadly speaking, the types of research that we do with animal models fall into three categories. So the kind of what you might call the blue skies basic research, advancing scientific knowledge, how animals behave, develop or function, maybe not necessarily with any applied objective. However, that really does underpin the second part, which is what I'm mostly interested in, using animals as models of human disease and to develop interventions. If you ask the general public, they're very, very, very supportive of this type of work, a lot less so of this type of work, and I think as biologists, biomedical scientists, we need to make the case more that really this underpins the work that we do in animal models to try and bring new treatments to bear on diseases. But there's also quite a significant part of animal usage which involves toxicity testing of pharmaceuticals and chemicals, including kind of environmental toxicity testing. That is a regulatory requirement. Actually, that's, you know, we have to do that testing. And you could argue it would be unethical to test a new pharmaceutical directly in human patients. And that's why it's a regulatory requirement to do certain tests in animals first. I've put this as a kind of fourth reason. It's not a main reason, but there is some animal experimentation that goes on in the UK that's regulated, that's actually for teaching. So surgical trainees, for example, may learn a particular technique, transplantation, let's say, on a pig, for example. So that's also allowed in the UK. Well, what about the ethical aspects? Well, I think maybe some may have argued a while ago that kind of what we do to animals doesn't really matter. They don't suffer in the same way that humans do. I think increasingly it's been realised that animals do obviously suffer pain and suffering that can be quite significant for that animal. And obviously many animals are killed in, virtually the vast majority of animals used in animal research are killed. So the question then comes up, I think that for me this is the crux of the kind of ethical dilemma. Is that justifiable? Is it justifiable for one species, in this case humans, to cause harm to another species primarily for the benefit of ourselves. And the kind of ethical argument has developed to the point where this I don't know if you've come across this term before, speciesism. Okay, so this is now a kind of recognised ethical position, and I guess what that would say is that to treat animals any differently from humans isn't appropriate. I think the other kind of perhaps a bit more relevant to what we're doing in terms of using animals as models of human disease is, does that produce useful results? Does it produce results that we can actually tell us something about what will happen for an experimental medicine in a human disease or not? Okay. So this whole idea of speciesism comes from the idea, or lack of, so the idea of speciesism comes from the idea that human interests are special in some way. Humans are different from animals. We can utilize them. We utilize them in many other ways. If you think about every sphere of interaction between humans and animals, it is exploitative, actually. So there are three, I think, main views. I think the perhaps traditional view would be that humans are morally distinctive, and some would still hold to that and argue that. There is a clear line view. We are clearly different and distinct from all animal species. The other view, and I probably lean more towards this, and I think the legislation leads more to this, there's perhaps a hierarchy of moral importance, so a sliding scale. So this idea that we're morally distinctive, if you argue, you could argue, for example, that the difference between an oyster, let's say, and a great ape is significantly more different than between a great ape and a human. So that's why some have argued that this distinctive line is arbitrary. The sliding scale kind of takes into account the neurophysiological sensitivity of the animal that you're talking about. So if you think in terms of biological complexity, you could argue humans are perhaps the most biologically complex and therefore have the greatest capacity to experience suffering and distress and joy and all those other emotions as well. And that ability to feel those emotions, to feel distress or joy is reduced as you go down the kind of evolutionary chain, let's say. The other kind of view is that there is absolutely no categorical distinction. So some of the speciesists, this is what they would argue. That we should treat animals, they're living entities, in the same way that humans are, and we should treat them in the same way. They have the same moral rights. So this idea of speciesism came from a guy called Peter Singer. So he's an Australian ethicist, and he wrote this book called Animal Liberation in 1975. And that's kind of the Bible of the animal rights organization. That's really what got it going. So he follows what's called a utilitarian approach. So utilitarian ethics involves weighing benefits. And a utilitarian would look for the moral, so when faced with a moral dilemma or a moral choice, you would make a selection based on what would give you the greatest utility. In other words, the greatest good for the greatest number. So as I said, Singer came up with this idea that the boundary between humans and other animals is arbitrary, that they share capacity to suffer with humans, and that speciesism violates the principle of equal consideration of interests in the same way that racism or sexism would. So it's actually quite difficult to argue with. He's followed up with other books, and I think we have to accept to an extent that we are speciesist. But this has kind of fed into the legislation as well, this approach, this idea that we should consider an animal's interests, not necessarily equally with those of humans, but we certainly considerably to a greater extent than we have in the past. So in the UK, there's a law called the Animal Scientific Procedures Act. And this was, for a long time, considered to be, and probably still is, the most rigorous regulation for animal experimentation in the world. So it imposes quite a high burden of regulation on scientists conducting experiments on animals. It was revised to come into line with a European directive. Actually, this European directive brought the rest of Europe closer to what the UK legislation imposes, although there were a few additional things that this added in. So it is a criminal act. It can be enforced by prosecution through the courts. I'm not sure if anyone actually ever has been prosecuted. You probably would. You would actually be charged under, not necessarily under this act, but under the Animal Cruelty Act, which is a much older act. It's implemented by the Home Office in the UK. So so actually Secretary of State is responsible for granting licenses for animal work. There are 3 types of licenses. Project license, which has, as you can imagine, has an overarching theme that might justify a scheme of work, say for a particular grant. There's personal licenses, which license individuals, and then there's establishment licenses. So Manchester University, for example, will have an establishment license that allows them to house animals for experimentation and for experiments to be conducted there. There's also an inspection regime, which is quite effective. They do just turn up unannounced. They'll ask you what you're doing. They'll come into the labs. They'll go through your records. They'll check that you're doing everything correctly. There's a whole section on kind of housing and husbandry, so that the standards of care are similar, essentially similar, or perhaps even better than you would expect in a veterinary practice, and that's kind of the standard that's applied. So quite a substantial regulatory burden, but for our perspectives, so just to say first that it uses this utilitarian approach. So I think it's the sliding scale view. And what it says is that the benefits to animals and humans, because of course animals do occasionally benefit, although from research on animals with new treatments and so on, but the benefits to humans is obviously considerably greater. Those benefits must outweigh the costs or harm to any animals. And the interesting thing here in terms of benefits is they don't necessarily have to be tangible. So they need to be the potential or the perceived potential future benefits, which is, I think, is a bit of a difficult one to quantify when we're doing research. It also imposes ethical review of licenses, so you have local ethical committees who will review licenses. Ultimately, the decision to grant a license will be taken by the Secretary of State. He'll be advised by the Home Office license inspector for your area, who will have gone through all of the paperwork and so on. But this is kind of the key thing from our perspective, thinking about data and how we generate data and the kinds of experiments we do. So this is actually the wording from the Act. So paragraph 5. So it says, the Secretary of State shall not grant a project license unless he is satisfied that the purpose of the programme to be specified in the license cannot be achieved satisfactorily by any other reasonably practicable method not entailing the use of protected animals. So that means if there was a case that you could obtain your scientific data, or you could achieve your scientific objective without using animals, then there's a legal requirement for you to do so. You have the burden is on you to prove that you need to use animals. So this is sometimes called replacement. Here in paragraph B it says, the regulated procedures to be used are those which use the minimum number of animals, involve animals with the lowest degree of neurophysiological sensitivity. So if you have to use animals, you use those lowest down the scale of neurophysiological sensitivity. So for example, you might want to use a fish before you would use a mouse before you would use a primate, and that needs to be justified. Cause the least pain, suffering, distress, or lasting harm, and are most likely to produce satisfactory results. All very difficult things to evaluate. There's detailed guidance on how we should go about this. But certainly the guidance relating to using the minimum number of animals puts a requirement on people to make sure that they're doing proper experimental design before they actually start an experiment. And actually it perhaps wouldn't be a bad idea for the rest of biomedical science to do the same thing. So we have to do some kind of power analysis. We have to justify the number of animals that we're using before we start. So that's kind of a legal requirement. And these two are called, so this is called reduction, using fewer animals, and refinement, using the least harmful methods. So those are called the 3 R's, reduction, refinement, replacement. And they've been around a while. They were first introduced by these two lovely chaps here, Russell and Birch, in 1959. So they were kind of pro-animal rights. They recognised that animal experimentation wasn't going to stop, that we did the work, there was a case for doing animal experimentation. And what they advocated, and they were ahead of their time really, was that if we're going to do it, let's do it in the best possible way that we can. So now the kind of, and this now is incorporated in law, as you've just seen. So we must replace, we must reduce, we must refine. And there is actually a funding council, the National Council for the 3 R's, NC3Rs, and they will fund work to either replace animal methods with non-animal methods, refine or reduce numbers, and so on. And I've never actually looked at whether kind of new analysis methods have been funded, but it wouldn't surprise me if they had. They also fund fellowships, that kind of thing. So there's a legal imperative, there's funding streams there, and it has become, over the years, a kind of increasingly important aspect of the animal research agenda. And certainly as a researcher, you want to be able to argue that you're using the best possible methods, that you're only doing things that are justifiable, and in a way, this gives us a framework for that. So that's like a bit of a background on the ethics and the legal side. So I'm going to talk about our mouse models of motor neuron disease. So I'll talk a little bit about the human disease. So motor neuron disease is a very simplified view of what motor neurons do. Here's the brain. In the motor cortex, you have upper motor neurons, so the cell bodies in the motor cortex. They send an axonal projection down into the brain stem and into the spinal cord, where they synapse with lower motor neurons. Again, here's another cell body in the spinal cord. Then sends an axon out that joins a nerve, innervates your muscle, and that allows you to control muscle contractions, voluntary and involuntary. So what happens in motor neuron disease, which is an adult onset neurodegenerative disease, you get degeneration of these critical cells, okay? And that leads to muscle atrophy and eventually death. And the life expectancy is very short, around 2.5 years from diagnosis. There is one drug, but it only prolongs life by, on average, 3 months, although some people maybe respond better than others. There's probably subgroups of the disease with responders and non-responders. At any one time in the UK, there's about 5,000 people with the disease. That doesn't sound like a lot, but the reason for that low prevalence is this very rapid progression. It's described as the worst disease in medicine by many doctors. Lifetime risk is less than 1 in 100,000. So if you lived, so for example, multiple sclerosis, there are a lot more people alive with multiple sclerosis. If your life expectancy with MND was similar to multiple sclerosis, you would probably have a similar number of people. The prevalence would be the same. So clinically, mainly the consequences are on motor function, as I've already described. There are some cognitive changes. So some of the neurons in the brain that are involved in cognitive processing can degenerate as well. That's been increasingly recognized now. But for example, he's showing here significant atrophy of the muscle that's innervating, the muscle that's controlling the thumb here, respiratory muscles that control obviously your diaphragm, breathing, lower limb, and the bulbar muscles that control speech, swallowing, and so on. And the main thing that patients die of is respiratory insufficiency, so they can't get rid of this carbon dioxide from their blood. They go into a coma and then die. In terms of genetics, things are being built on all the time, actually, and this is a little bit out of date now. There are quite a few genes associated with motor neuron disease. These are the main ones we're interested in for reasons hopefully I'll explain in the next few slides. Mutations in superoxide dismutase 1 were the first to be identified back in 1993. If you look at graphs of when genes have been identified, it's absolutely skyrocketing at the moment. One of the latest ones to be identified was a gene called C9orf72. That's responsible for about half of inherited motor neuron disease and about 13% of apparently non-inherited disease. TARD-BP is a protein called TARD DNA binding protein of 43 kilodaltons, or TDP43, is quite interesting. Mutations are found in a small number of patients, but I'll show you in a minute the pathology involves this protein, and that's present in the vast majority of patients, so we're interested in that particular gene as well. But it's important to remember that with all these genes, only about 5 to 10% of motor neuron disease has a genetic cause. And the rest is what you call apparently sporadic. It's just arose spontaneously. And so that's one thing to remember, is that all of the models that we use are based on kind of mutated genes. There are risk factors. Age, so the older you are, the higher your risk. Males have a higher incidence than females. Physical activity is a risk factor. That's no excuse not to do any physical exercise. Your relative risk is increased, but it's still a relatively low probability overall. There was a study of Italian footballers between 1970 and 2000 saying they had a really high risk, but actually they identified 5 footballers with ALS, whereas there perhaps should have only been less than 1. About the predicted number of cases would have been 0.7. But this has often led to the idea that physical activity, trauma to the head, other things like perhaps drug usage may have contributed to motor neuron disease, or this could just be a bit of an outlier. These kind of epidemiological studies are very difficult, probably as you know. So there have been some that have associated MND with increased risk with physical activity, others not. There are some very famous sportsmen. So this is Lou Gehrig, and it's sometimes called Lou Gehrig's disease in the US. He was a baseball player, and it's very interesting. You see his batting averages plummet before he's diagnosed with motor neuron disease. So you can have significant loss of muscle before you actually are diagnosed. I gave this talk in Leeds once, and no one knew who this guy was. Don Revie, so he's Leeds manager. He was diagnosed with motor neuron disease as well. So disease mechanisms, virtually everything that goes on in a cell has been linked with motor neuron disease. And I blame most of this on kind of microarray analysis. So obviously you pick out your favorite biological pathway. Although there is some good evidence for some of these, and with the number of genetic causes of motor neuron disease that are increasing, we're getting a better handle on this, I think. So processes where RNA, involving RNA processing, transport, splicing, export from the nucleus are looking quite interesting at the moment. So I mentioned earlier this protein, TDP43. So these are, and this is characteristic of the pathology in virtually all patients. So these are spinal motor neurons in the spinal cord. So this is the motor neuron cell body, massive cells, that's the nucleus there. And ubiquitin is a protein that's tagged proteins that are going through destruction, and you get these accumulations of ubiquitin when that process fails, and it just starts to kind of just store big clumps of protein inside the cytoplasm. So this is pathological. You get these kind of skein worm-like inclusions or compact inclusions. So they stain for ubiquitin. And then in about 2006, it was realized that actually there's another protein in here called TDP43. And that's a key protein involved in RNA regulation. Does all kinds of stuff. The interesting thing is that patients, the models we've mostly been using, because SOD1 mutations were discovered first in 1993, we've been using models, and the model I'm going to talk about is one based on SOD1 mutations. Patients with SOD1 mutations don't have TDP43 in their ubiquitinated inclusions, so there's quite a big difference between patients with SOD1 mutations and all other patients. So even patients who don't have mutations in TDP43 have TDP43 in inclusions. It's worth saying this is at end stage, so all of this is from people who've died with motor neuron disease. So this could be an end stage protective mechanism actually. And there's some evidence from the animal models that may be true. Some of the animal models based on TDP43 don't necessarily show this early on. So this kind of comes back to the kind of face validity of our animal models. If we're going to argue from an ethical perspective that this data is valid, then we need to be able to say that our models are similar to the human disease. So the kind of ideal characteristics of a preclinical MND model, I won't go through them all, are kind of listed here, and they're based on a similarity to the human disease. So if we look at, these are the 3 main models that we have in Sheffield. So we've got models based on SOD1 mutations, models based on TDP43 mutations, and models based on C9orf mutations, so mice that are transgenic for the human genes with these mutations in. Then they, to a greater or lesser extent, satisfy the different criteria that we're interested in. Part of the problem here is we don't really know for some of these models, even though they've been around for a while, they haven't been systematically evaluated, and that's something we're trying to do at the moment. If I've got time, I'll show you some data on the TDP43 model. We've just got hold of a model that's got a C9orf72 mutation, and we've got no idea actually at the moment whether that's going to be a valid model or not. But this also comes back to the idea that actually if we rely on one model, that's probably not as valid as being able to assess new therapeutics in different types of familial motor neuron disease. And ideally, ultimately, we're going to be treating patients with sporadic disease. So we need to have some way of understanding whether sporadic disease is modeled by any of these different approaches. I think this model here, based on the pathology that we see in humans, models based on TDP43 mutations might have the greatest potential for that. But I'm going to focus on the SOD1 model, and I think it's interesting, illuminating for some of the big problems that people have had with it, which I'll talk about in a moment. But first of all, just to show you what happens in those mice, this is our data. So this is just a graph showing age in days versus kind of neurological score. So that's a measure of neurological deficit, how paralyzed the mice are. And there's a distress score as well. So as part of the kind of ethical process, we had to introduce a system where we could measure the distress in mice. And you might think that's very difficult, but actually it's really easy to do. Once you're skilled at it, it's very easy to tell whether a mouse is in pain or distressed. And that's what this score takes into account, as well as their kind of level of paralysis. So they start with disease, obvious signs of clinical disease that you can see about 75 days. They have tremor, hind limb splay. They progress to kind of a spastic, waddly gait, kind of a swimming gait, and then they move on to have more kind of frank hind limb paralysis. Perhaps one limb will be significantly paralyzed. In terms of the distress score, though, actually they still seem essentially not really very distressed, despite having a high level of motor dysfunction, particularly in the hind limbs. They then progress on to significant hind limb paralysis, begin with some forelimb weakness, and then they reach an end stage, a humane endpoint, which is a complete loss of the ability to turn over within 10 seconds. So if you put the mouse on its back, it can't right itself within 10 seconds. And you're interested in you talking about survival analysis earlier. So people call that survival time. And they use Kaplan-Meier plots to analyze that data. But I'm not actually sure that's right, because 100% of our animals reach that endpoint. So I've had some statisticians argue to me that if that data is normally distributed, there's no need for us to use kind of survival analysis, because all of our mice reach that endpoint. So the distress, as you can see, is really kind of concertinaed in the last 10 to 14 days of the disease course. Okay, so that's an important one to remember for later. But the big problem with this model that's been prevalent in the literature over the last 5 years or so, 10 years, is that this is what people say about the SOD1 transgenic mouse model. The results in it don't translate to the clinic. And actually, you could go to pretty much any disease area where people are using mouse models or animal models, and they'd say the same thing. And I've not got time to go into why all of those, why that might be, but I think there are very good reasons why that might be. Experimental design is a big one. I'll talk a little bit about that now. So many compounds have apparently worked in the SOD1 mouse model, and they have not then worked in the clinic. So it could be because, as I said, the SOD1 model doesn't represent the rest of motor neuron disease. That's certainly feasible, possible. We've got other evidence that it does. There could be technical issues, so poor study design, variable model, publication bias. There's certainly evidence for all of that in the model, and I'll talk about that a bit more. And there's also what's called the translation pathway. So when you go from a result in a mouse to a clinical trial in human, there's some very kind of critical things you have to do to make sure that you give yourself a good chance of seeing the same effect. If you look at all of these drugs, in fact, every drug that's failed in motor neuron disease, there's been kind of key steps in that translation pathway that have been missed or ignored or not appropriately addressed. But just thinking in terms of the actual mouse model itself, what are these study designs' technical issues? So there was an institution called the ALS Therapy Development Institute. It's a kind of not-for-profit biotech company set up by a patient with some influential, wealthy benefactors as well. His family was quite wealthy. And they just got some of these mice and started throwing drugs at them and seeing how long it took them to reach that end stage, the kind of survival time, as it's called. How long does it take the mice to reach end stage disease? They did loads and loads and loads of studies, and they weren't really getting any results. And then they went back and looked at all of their data. So they tested about 70 compounds in 221 studies, about 18,000 mice, about 5,000 of them are on control, so not given an experimental treatment. And they've got all of that, I think, in a database. They've certainly got the control mice in a database. And what they did was they then modeled, and I don't know whether it was an appropriate way of modeling or not, but they just went into the database and said, if we just randomly select groups of a particular N, what's the chance that we just pull out random differences when there's actually no fundamental underlying difference? And the frequency, the probability was incredibly high. So something like 40% of the time, 50% of the time, maybe higher, they'd just come back with a significant difference, well, not a significant difference, a kind of more than 10% difference in survival, where in fact there shouldn't have been any. And they identified some of the issues around this model, which were that people, if you don't censor kind of motor neuron disease, ALS disease-related deaths, then you have an issue. So sometimes mice might die because they have a dosing injury, they have a reaction to the drug, etc. You need to censor those deaths. That was one of the first things they identified. The other major thing was that they needed to litter match. So when they had a litter of mice, you need to take pairs of mice, put one on the treatment arm, one in the control arm. When they did that, that significantly reduced the variability. The other thing was they found that the kind of number of mice that you needed to use was much higher than everyone else was using at that time. So they use about 24 mice per group, 12 males, 12 females. And when they did all that, they could really reduce the variability in their system or increase the precision with which they could detect effects based on kind of probing this database. And then they retested lots of compounds that have been said to be positive in the literature, and they were all negative. So these are the original published studies that they tried to replicate with their new optimized design. These are the percentage extension in lifespans that were reported in the literature, quite often in, as you can imagine, high-impact journals. And when they repeated with their balanced kind of appropriate censoring, litter match controls, protocol, they got absolutely no effects with any of them. So it caused a bit of a stir in the field. They had trouble getting it published, actually. They did eventually get it published, and since then it's been quite influential. So this led to the Scott guidelines, and it's published here. There's since been generic guidelines, because a bit more generic guidelines, because this model is unique. This model basically is genetically ill-defined. Okay, so most mouse models that we use are very clean genetically, as it were. So they're highly inbred, and they've been inbred for multiple generations, so they are homozygous at every genetic loci. So essentially a daughter, son of a parent looks essentially genetically identical to the parents. That would massively reduce his variability, and I personally I think that's a very desirable thing to do when you're trying to establish relatively small effects of experimental treatments. This model, for some reason that I still haven't fathomed, they had a mixed background from the beginning. So they crossed two mixed background mice together, which leads you into a genetic land of kind of chaos, really. And most of the variability comes from that genetic variation, a huge amount. So what we did, we did this before all of this stuff came out. We back-crossed these mice onto a congenic background, C57 Black 6 background, okay, for more than 20 generations. And immediately we kind of halved the standard deviation of the time to reach end stage, which is a massive reduction. And then we developed a range of kind of objective measures of disease progression. The other thing was we looked, and I'll show you in a moment, we looked at other measures of disease progression, not just throw a drug and see how long it takes for the mouse to get to a very severe stage of disease. So we published that protocol, but just to go through some of our data. So one of the first things we did, and I was saying to Neil earlier, we need to get all our data onto some kind of sensible database so we can do this much more quickly, because this is also out of date. So these are 6 studies over about 2 years, 6 different studies, and it's showing the time to reach this end stage disease for the 6 different studies, and also the Rotarod performance. So what this is you put the mouse on a rotating wheel and you accelerate the wheel, and eventually they fall off, and you measure how long it takes them to fall off. It's quite a crude test, and it's very, very variable for individual mice. When you average across a relatively large group, it's not too bad. But hopefully what you can see is across a whole range of different treatments, these are all the placebo groups from a range of experiments, you get essentially the same profile, a massive reduction, kind of leveling off, and then another significant reduction in Rotarod performance. The end stage, again, is pretty much bang on from experiment to experiment, and we could extend this now. I think we've probably done about 15, 20 studies, so we should probably do that. But the average survival is always 140 plus or minus 6 days, and we always see this kind of Rotarod profile as well. I'll just show you from that Scott paper, and sorry, the quality's terrible. This was scanned from, this is the scan the British Library sent me. So these are their however many studies, thousands of mice. So this is their same data, but you can see the spread is actually huge. For the ones that they think are the kind of the real deaths here, these ones are due to a genetic loss of the transgene. These ones are deaths due to other reasons, not ALS. So the spread is massive. So if you just go back to ours, it's kind of much tighter. We can get away with much smaller numbers to show the same level of effect. So what this led to was, so this is just showing that Rotarod data again as a kind of average across several experiments. So the interesting thing here is that we're losing Rotarod performance at a stage before the mice look like they have disease. There's a massive loss in Rotarod performance, about a 40–50% loss in Rotarod performance at an early stage. And we can link this to changes that are going on in the muscle that we know about now. So the motor neuron axons are detaching from the muscle, and the muscle begins to degenerate quite early. But you don't necessarily see that. The mouse doesn't move differently. If you put a normal mouse onto a Rotarod, it consistently scores about 300 seconds on this test. So we thought that actually that provided a massive window. We don't need to take animals through to a very substantial phase of the disease to measure the time it takes them to reach the end stage. We could just measure whether they kind of drop off quickly or not. And that's really the basis of the work that we've done to kind of refine this model and reduce the number of animals that we use. So we believe now that, and I'll show you some more data, that we can test therapies in this initial phase where we're seeing a very significant loss in the innervation of hind limb muscles early on. And then if we can see that, then we could go on and look for perhaps changes in later phases of the disease. So the other kind of quantitative measures that we introduced were magnetic resonance imaging of muscle volume. So these are normal mice. These are the SOD1 G93A mouse model of motor neuron disease at 30, 60, 90, 120 days. And this is a kind of section through the lower limb muscle. So you can see a massive reduction in muscle volume, and that's quantified here. And again, that's between 30 and 60 days of age, quite huge differences. And this is quite a nice technique because we've got quite low coefficients of variation. This is a bit harder to do. It's not really very high throughput, but we can measure how many, these are neuromuscular junctions, this kind of pretzel-shaped structure is the neuromuscular junction. The green is the axon coming in that's controlling this muscle. And you get loss, as I've said, of that axon coming into the pretzel shape. So we can measure that loss of innervation, which again is huge. This is at 60 days of age. We can also do, we also thought this would be quite interesting to look at gait analysis. So we've got a system that can kind of record their foot patterns, their footfall. So it's just a glass catwalk, and the mouse runs across, and then you have a video camera underneath. So there's a kind of software associated with that. It's not particularly high throughput again, because you physically have to, so the software tries to recognize the foot patterns, but it can't, it fails quite often, so you have to go in and correct it. So the students always complain when they've got a lot of catwalk analysis to do, because there's a lot of manual curation and labeling of which limb is which. But it is quite powerful. So that's a normal mice, that's a mouse with motor neuron disease. So the red is right, green is left, and then the forelimb is bright, the hind limb is dark. And you can see a normal mouse will run, and its hind limb will be placed where its forelimb was, which is what's happening here. These mice have a much shorter stride length, and they're also quite, as you can see, they're staggering around a bit. And there are a huge number of parameters that you can measure from this, some more useful than others. But we don't really see this change very early on. So what this is measuring is kind of the spastic gait, the quality of the gait. It's not measuring power. So the Rotarod test, we think, is measuring power. So that's showing differences quite early on. This shows some differences much later. So how long they spend on one particular limb. Their stride length also reduces. the amount of time that they're supported on diagonal limbs goes down, whilst the time they spend supported on 3 limbs goes up. So again, quite useful quantitative data rather than just looking at the mouse and saying, yes, that's a clinical score of 1 or 2. This is a bit more objective. So that led to this kind of study design where, as I said earlier, instead of taking mice all the way through to end stage, we do a whole range of tests up to about 90 days of age. And I'll show you later, we've since expanded on this. But the Rotarod test is the most variable thing that we use. If you look at plots for individual mice, it can be all over the place. So we normally reduce that to a single parameter for each mouse. So we measure the time it takes for a mouse to reach a 20% decline in Rotarod function. So quite a crude single measurement, but that makes the analysis much more robust in our hands. There may be better ways to do it. But we wanted to try and improve on the Rotarod test itself. It's also, you have to get all the animals out of the cage, put them on the Rotarod. It takes a long time. It kind of stresses the mice and so on. Well, some people in the lab were looking at this system to exercise mice. So you put a running wheel in the cage, and we thought this might be a useful measure. You put a running wheel in the cage, it's just a kind of round disc. It's at an angle, and it can rotate, obviously. It's got a magnet, and then there's a reed switch, which this is just a bike computer stuck on the outside of the cage with a bit of blue tack. So you can measure kind of average speeds, distance ran, maximum speed, which actually isn't quite that useful, and so on. And this is nice because the mouse is in the home cage. All we need to do is come in and read it off. We get data, much higher density of data. We get data every day. And the mice really enjoy running on running wheels. So this is, I think this is a normal mouse. It's not a G93A mouse. So they just run and run and run and run. Anyone want to guess how long they run for in one night? Anyone? Throw a number out. Number of hours? 3? Any higher? 5's not bad. Distance? It's quite a long way. So, I mean, to be honest, we should be careful a little bit about interpreting this data because as you'll see in a moment, they don't always run. Sometimes just play on it. So you'll see in a moment it'll stop and just whiz round a bit. And some mice do that more than others. So some people have argued this is a stress response. They're stressed, that's why they're running. But there's I need to find the paper. I always say this when I give this talk. There's apparently some Dutch guy, researcher, put some of these wheels in his garden and filmed wild mice getting on the wheels and running pretty much just as much as these mice. So we think it's a normal response. No, I need to find out. So this is average data for normal mice. So I think they were about 30 days of age when they were put in. So this is time from introduction to the wheel. So there's a learning phase. So they take a while to get used to the wheel. Some mice don't run. Some mice just won't run. There's a much higher frequency of males that don't run than females. It always pains me to say that. So we actually use female. There's various other reasons why we use female, but in our experiments we always use female mice. They run much more with a much higher frequency. So the time is about at peak, so there's a kind of plateau phase here. It does tail off with time, probably about 5 hours a day, or a night really. And the average distances we've calculated is 15 to 20 kilometers during that peak. Average speed is quite nice. So we are doing some work with one of Neil's students, I think. Alan, yes. So we're not getting all of the data from this. So the people who originally developed this methodology did hook them up to data monitors that would collect data in much shorter time bins, and they showed that they were cruising. The mouse had a cruising speed, and that was actually much more a robust measurement of activity. So Alan's he's developed a device to actually record continuously from these running wheels. So we've got with that much more data now, and we hope to. The idea will be if we can develop that further, we're going to get much nicer measures out of the data than we're using at the moment, which are a bit crude, but they're not bad. So that's a normal mouse. So that's a SOD1 mouse. And hopefully you can see this is the kind of statistics I like, loads and bugger all. So they train up from the age of about day 30. They don't quite get to the same peak, but then they almost immediately just drop off again. So on all three measures, maximum speed is a bit useless really. We should probably stop showing that. Because if they change direction, when the magnet's close to the reed switch, you'll get a very fast maximum speed. We think that's why. So we did very basic statistical power analysis on the time taken to reach a 20% decline in either running wheel performance or Rotarod performance. And the Rotarod test would need about 14 mice per group. This would take about 5 mice per group. In practice, we don't run 5 mice per group on this fast track methodology. We use more. We're normally doing multiple groups. But it does open the possibility of doing kind of more screening, much more kind of larger scale screening type experiments, which I'd be interested in exploring. So with small numbers of animals per group. And it's quite reproducible. So that's six different studies. They're all showing essentially the same thing. That's all of them kind of superimposed. And this is for a kind of in vivo, bloody thing, in vivo readout. I think this is really impressive. So the between study coefficient of variation is 3.9%, which is, I think, super impressive. The other nice thing about this is that riluzole works. So riluzole, the drug that works in patients, works in this system. It doesn't change the time to reach end-stage disease, but it does slow down this decline. Not particularly massively, but it does slow it down a bit. So that's quite nice to show that. And now we've got a whole range of kind of methodologies. So the other way we try and avoid kind of false positives, which still are possible, I think, it's certainly feasible, is to have a kind of staged approach. So we might use the fast track running wheel to begin with. If that works, we then go on to a more kind of looking at Rotarod performance and onset of disease up to day 90. With these methodologies, we can take tissue at different time points and analyze what's going on at a pathological level. But then eventually we might actually, if these two work, move on to a study where we look at the survival or time to reach end-stage disease. I shouldn't say survival, really. So I think using that kind of approach, we build up a picture for a particular therapeutic agent of whether it's effective or not. I don't know, do you want me to stop there? I could. Yeah, okay. So, well, I was going to talk, let me just show you a video of, so we've got this other model, the TDP43 mouse model. Sorry, these videos don't come out very nicely, but, so this is a normal, they're quite fat, these mice. So that's a normal gait. It looked quite waddly, but I don't know if you can see the difference between that and that, where it's having difficulty keeping its kind of hindquarters off the ground. I'll just play it again. This model's interesting because, so the mutant mice show a neurological phenotype. Mice expressing the mutant TDP43 genes show a mutant phenotype, but the mice expressing a normal TDP43 gene don't. And it doesn't progress to as severe a level as the SOD1 model does. So we're quite hopeful. We've got a PhD student working on this. We've got some quite nice data out for some of those other measures that I've already talked about. So quite hopeful that we'll have two systems that we can check out therapeutics in. Okay, so that's me.